hypothesized that CAFs may function in promoting immune response via communication with immune cells. And then the final insight that we can gather from this analysis is that it might be important to differentially target stromal cell subtypes to remediate tumor growth in triple-negative breast cancer. So, for example, we may want to downregulate pathways in PVLs if we hypothesize that they contribute to tumor growth. And on the other hand, we might want to upregulate pathways in CAFs in order to promote immune response to target tumors. And then finally, to summarize this talk, so we were able to cluster stromal cells in triple-negative breast cancer into CAF subtypes and PVL subtypes. These clusters were also found to be spatially distinct, with PVLs localizing towards blood vessels. And we also hypothesized that PVLs may contribute to tumor growth via the VEGF pathway, while CAFs may promote immune response in tumors instead. All right, and that concludes our talk. So I can move on to the Q&A section now. If there are no questions, I believe my colleague has also sent over a poll for you guys to answer as well. We'd be interested in seeing like which talks you guys want to hear next, which platforms you guys are interested in seeing in these talks. Okay, so if there are no other questions, I guess we can finalize the talk here and end here. Thank you guys for joining as always, and we hope to see you guys. Oh, I guess we have two questions. Okay, so the first question is, can you show again how you perform analysis in one patient? So yeah, I can tell you how I subclustered the data set. Let me open up my data. Okay, so this is my data. To find the subcluster, I can open up the different type of metadata that I have. For me, it's under subject ID, which is all of the 5 patients. And you can choose one patient, for example, and click subcluster. So subcluster is basically subsetting the data. Oh, sorry, I have to select this and then subcluster. Right, and when you subcluster or subset the data, this does not reperform the UMAP or any clustering in any sort of way. It kind of just zooms in on that subset. If you want to perform the clustering from here, you'd have to reperform the Louvain clustering. Okay. Okay, we have another question from Kristen that asks, could you explain a bit more on how you got to the network graph? So I'm assuming this is on cell chat. Okay. So let's say we have our data set here. For me, I ran the analysis with just all default settings because I just wanted to play around with cell chat and see, what I can gather from the results. So here you can type in the analysis name. You can type in your database here. We have human, we have mouse. Okay. Then we also have different ways of computing the average gene expression as well. Okay. Yeah, but basically I just ran with all default parameters, and then clicking this results page, it takes me to this page right here. Okay, and there's different types of pathway analysis that you can perform. If you click new pathway analysis, you can specifically select your pathway here that you want to enrich for, and then also specifically select the receivers. that you want to look at, right? And this makes it so that you're not having a lot of information thrown at you at once. But yeah, after that pathway is run, you can flip through your different pathways here. Okay. So I hope that answered your question. Great. Okay, and then another user asks, how can you determine the root cell in pseudotime? Okay, so in my case, let's go back to the slides. Okay, so here typically you want to have a hypothesis in mind already. For me, it was quite easy because I only have two cell subtypes that I wanted to look at. So I wanted to see if, iCalves differentiated into MyCalves in some way or vice versa. But if you were to have multiple cell types, I would say choose the root cell that is kind of on the border of that cluster in order to see the full trajectory of it. Of course, you can rerun this analysis using different root cells to see, if that would change your biological insights in any way. But yeah, Bioturing would basically recommend the root cells for you to see what results you would want to get. Okay. And then another person asks, how do you perform the pseudotime analysis? So going back here, if you have your study open on bbrowser X, the pseudotime analysis is on the left-hand side right here. You can just click that button. And then it'll prompt you to create the principal graph, which is basically asking you what type of metadata you want to include. And typically you want to include the clusters, right? And since we have already annotated the clusters in this step, I wanted to include these two clusters specifically, right? And after you choose that metadata, it'll take you to this screen where you can then choose your root cell. And then after you have chosen your root cell, then you can click run and it'll show you a results page similar to this, where you can look at the pseudotime and you can query for different genes to see if it would, increase or decrease in any way. Okay, and I think that covers all of the answers. Yeah, so thank you guys for joining, as always. There are no more questions. I can finalize the talk here. All right, thank you, everyone.